For our honors project, we went and visited the Field Museum in Chicago, known for its exhibit on evolution. For our project, we were asked to create a video podcast on the evolution of a certain topic. For our topic, we chose the evolution of gene sequencing.
Gregor Mendel is known as the father of modern-day genetics. He used a monastery garden for crossing pea plant varieties, having different heights, colors, pod shapes, seed shapes, and flower positions. Mendel's experiments between 1856 and 1863 revealed how traits are passed down from parents. For example, when he crossed yellow peas with green peas, all the offspring peas were yellow, but when the offspring reproduced, the next generation was 3/4 yellow and 1/4 green. Mendel's work showed that what we now call genes determine traits in predictable ways.
When Francis Crick and James Watson modeled the structure of DNA, they used paper cutouts of bases and metal scraps from a machine shop. Their model represented DNA as a double helix, with sugars and phosphates forming the outer strands of the helix and the base pointing into the center. Hydrogen bonds connect the bases, pairing A to T and C to G, and the two strands of the helix are parallel but oriented in opposite directions, 5' to 3', 3' to 5' the other way. Later on in the early 1960s, Marshall Nirenberg and the National Institutes of Health colleagues focused on how DNA directs protein synthesis and the role of RNA in these processes. Their 1961 experiment using a synthetic messenger RNA strand that contained only uracil yielded a protein that contained only phenylalanines. Identifying UUU, 3 uracil bases in a row, as the RNA code for phenylalanine was their first breakthrough. Within a few years, Nirenberg's team had cracked the 60 mRNA codons for all 20 amino acids.
In 1977, Frederick Sanger developed the classic rapid DNA sequencing technique, now known as the Sanger method, to determine the order of bases in a strand of DNA. Special enzymes are used to synthesize short pieces of DNA, which end when a selected terminating base is added to the stretch of DNA being synthesized. Typically, each of these terminating bases is tagged with a radioactive marker so it can be identified. Then the DNA fragments of varying lengths are separated by how rapidly they move through a gel matrix when an electric field is applied, a technique called electrophoresis.
In 1983, Kary Mullis discovered the polymerase chain reaction, which is a relatively simple and inexpensive technology used to amplify or make billions of copies of a segment of DNA. One of the most important scientific advances in molecular biology, PCR amplification is used every day to diagnose diseases, identify bacteria and viruses, and match criminals to crime scene. PCR revolutionized the study of DNA to such an extent that Dr. Mullis was awarded the Nobel Prize in Chemistry in 1993. There are 4 major steps for running PCR. The first step is simply collecting the DNA sample from the